Hello everyone. Yesterday we have discussed the endoplasmic reticulum and its parts. Today we are going to discussing Golgi apparatus, that is a most important feature of cell organelles, because without Golgi apparatus there is no sorting, packaging and transportation of proteins will be occurs. So we have discussed the structure of Golgi apparatus later. First we have take a glance on the specific features of Golgi apparatus. So Golgi apparatus, also known as the Golgi complex, Golgi body, and also known as the cellular shipping and processing center. It is an important organelle in eukaryotic cells which is responsible for processing, modifying and packaging proteins and lipids received from the endoplasmic reticulum. It ensures proper sorting and transportation of these molecules to their designated location within the cell or for secretion outside the cell. If we think about Golgi apparatus as a postal sorting center where packages in form of protein and lipids are labeled, modified and sent to their final destinations. Without Golgi apparatus, cells would not be able to distribute proteins and lipids efficiently, leading to malfunctioning in biological processes. Next is structure and of Golgi apparatus. This is the structure of Golgi apparatus. Golgi apparatus is composed of a series of flattened membrane-bound sac called cisternae. This is cisternae. These are a series of flattened membrane-bound sac called cisternae, stacked like pancakes. It is polarized, meaning it has different region that performs different or distinct functions. So let us discuss about this is cis-face and this is trans-face. These two phases are very important in the structure of Golgi apparatus because these two phases are responsible for doing all the function for Golgi apparatus. Cis-face receive the newly synthesized proteins and lipids from the rough endoplasmic reticulum. So cis-face is also known as receiving site, while the trans-face, this is lower side, trans-face is situated, as the name indicates, trans means transportation or shipping occurs. The final and modified molecules are packaged into vesicles and sent to their final destination. And this is medial region, means processing zone. In this, protein and lipids undergo modifications such as adding sugar molecules to form glycoprotein or glycolipids. So this is cis phase, receiving phase, this is trans phase, this is shipping phase, means the material should be shipped out for the distinct place for their destination where it has to be targeted. And these are the incoming transport vesicles, and this is newly forming vesicles because the overall function is also done by vesicles. Lumen, the hollow structure, this lengthy structure is known as lumen. So this is the structure of Golgi apparatus. Now we will take an example, Golgi apparatus as a warehouse. Warehouse analogy means imagine a warehouse where the raw materials arrive, means Golgi apparatus is a place where the raw material arrived, and in this the raw material is processed, labeled and packed into boxes before being shipped to stores. Similarly, Golgi apparatus ensure that biomolecules reach their correct location. So this is an important example. We have to understand the basic function of Golgi apparatus. Now next is the functions of Golgi apparatus. There are so many types of function of Golgi apparatus. The major ones are processing and modifying proteins, which are in form of raw material. So protein synthesis in the rough endoplasmic reticulum are transported to the Golgi apparatus, where they undergo further processing, modification and sorting of their final destination. Modification include folding, cutting and adding sugar groups, means glycosylation. Some proteins also receive phosphate or sulfate group for activation. Next is lipid and carbohydrate synthesis. The Golgi synthesizes glycolipids and lipoproteins, which are essential for cell membrane and communication. It also produces polysaccharides, which is required for cell wall formation in plant cells. Next is sorting and packaging molecules. The Golgi apparatus sorts proteins and lipids into specialized vesicles that deliver them to their appropriate cellular or extracellular destinations. Vesicles, as I already stated in the structure, that this is incoming transport vesicle and this is newly forming vesicles. Means in newly forming vesicles the material should be intact and this transport vesicle is ready to go their final destination. So this is newly forming vesicle where the sorting material will be packed and from this vesicle it will be transport. Now next is some molecules are sent to other organelles while other are secreted outside the cell. Next is the transport of secretory products. Cells that secrete hormones or enzymes like pancreatic cells depend on the Golgi apparatus to package and export these essential products efficiently. Next is formation of lysosome. As we all know that lysosome is important in digestion. The Golgi apparatus also generate lysosomes, which are membrane bound organelles containing digestive enzymes that degrade waste material and cellular debris. So these are the main functions of Golgi apparatus. First we will discuss the processing and modifying protein. Second we have discussed lipid and carbohydrate synthesis. Next sorting and packaging molecules. Next transport of secretory products. Next last is formation of lysosome. Now the next point which we will discuss, the Golgi apparatus and protein processing. Protein produced in the rough endoplasmic reticulum are not fully functional. The Golgi apparatus modifies and activates them before they are sent to their target location. Now we will discuss the steps in protein processing. First steps is protein entry. How protein enter in Golgi apparatus. Protein enter in Golgi apparatus through the cis-face, because cis-face is the receiving phase. So newly synthesized proteins from the rough endoplasmic reticulum enter the Golgi apparatus through the cis-phase. Next is modification. Medial Golgi, means this is the medial structure of Golgi complex. Here proteins are folded, trimmed and tagged with sugar molecules. This is the area where the proteins are folded, trimmed and tagged with sugar molecules, And next is sorting and packaging. And the sorting is packaging occurs in the trans phase. Here modified proteins are packed into vesicles. So these are the vesicles which are ready to deliver and sent to their final destination. Now let us take an example for that. Insulin processing in Golgi complex or Golgi apparatus. In pancreatic cells, the rough endoplasmic reticulum is responsible for synthesizing proinsulin. Proinsulin means the inactive precursor of the hormone insulin. The Golgi apparatus processes proinsulin into its active form, that is insulin, which is subsequently packaged into vesicles and secreted into the bloodstream. Next is the Golgi apparatus and lipid transport. The smooth endoplasmic reticulum synthesizes lipids which then enter the Golgi apparatus for modification and transport. So in this, Golgi adds sugar molecule to lipids forming glycolipids that play a vital role in maintaining the structure and function of the cell membrane. Packages lipid into vesicles for delivery to the plasma membrane or other organelles. Now we will also take an example for Golgis in nerve cells. So the role of Golgi in nerve cells. The Golgi apparatus helps produce myelin, a lipid-rich substance that insulates nerve cells and enhances signal transmission. Damage to myelin leads to neurological disorders like multiple sclerosis, that is called in short form MS. Next is the Golgi apparatus and cell communication. The Golgi is essential for cellular communication by producing signaling molecules such as hormones and neurotransmitters, modifying membrane proteins which are important for recognizing and responding to external signals. For example, Golgi role in immune response. White blood cells use the Golgi to process and release antibodies that help in fight against infections. Next is the Golgi apparatus in plant cells. In plant cells, the Golgi apparatus has a specialized role in producing cell wall components such as cellulose and pectin. Next is polysaccharide, which provide strength and support to plant structure. For example, Golgi and fruit ripening. The Golgi produces pectin which affects fruit texture. As fruit ripens, Golgi modified enzymes break down pectin, softening the fruit. Next is the Golgi apparatus and cellular transport. The Golgi body forms different types of vesicles to transport materials. So there are secretory vesicles, transport vesicles and lysosome. The function of secretory vesicles is to release hormones and enzymes like insulin secretion. In transport vesicles moves protein and lipids to organelles, for example membrane repair. Lysosome contain digestive enzymes and breaking down bacteria. For example, Golgi role in digestive enzymes. The Golgi packages digestive enzymes into lysosome, which will help in break down food in stomach. Now the Golgi malfunctions and diseases. Defects in Golgi can lead to severe cellular dysfunction and diseases. First is congenital disorder of glycosylation. CDG is known in short form. A genetic disorder where protein do not get proper sugar modification, leading to neurological and developmental problems. Next disease is Alzheimer's disease. Faulty Golgi function may contribute to the accumulation of toxic proteins in neurons. Third is cancer. Dysfunction of Golgi apparatus can result in the unregulated secretion of growth factors, contributing to tumor development and progression. For example, Golgi function in Alzheimer's disease. In Alzheimer's disease, the Golgi fails to properly process proteins, leading to the accumulation of beta amyloid plaques which damage brain cells. Next is the Golgi apparatus in evolution and biotechnology. The Golgi apparatus has evolved to meet the complex needs of multicellular organisms. Its role in protein and lipid modifications makes it a target for research in drug development and synthetic biology. In drug development means understanding Golgi function helps scientists to design cancer treatment. While in synthetic biology, researcher aims to create artificial Golgi system to produce valuable protein for medicine. For example, artificial Golgi in biopharmaceuticals. Scientists are developing bioengineered Golgi system to produce therapeutic proteins like insulin and antibodies. Now lastly, we would like to say that Golgi apparatus is a cellular powerhouse. The Golgi apparatus plays a central role in cellular functions, ensuring that proteins and lipids are properly modified, sorted and delivered to their destinations. From protein processing to cell function, the Golgi act as a cell's shipping and distribution center. Understanding the Golgi is critical for biomedical research, helping scientists develop new treatments for genetic disorders, neurological diseases and cancers. Or we can say that without Golgi body, we cannot imagine that life will move smoothly or correctly. Thank you so much.